Well, I want to thank NSF for the invitation to speak to you today. We were a bit tentative when the invitation came in because of all the talks that you've heard today. We represent the youngest grant, and we've only been working on this project for about a year and a half, so we're kind of vamping up. But I'm glad that you did that, because we actually had to work on putting this talk together, and we realized how much we've already accomplished, and that's been great. And it also provides a great opportunity to actually get feedback from all the people working on dimensions of biodiversity, and maybe provide some additional things that we could do that we didn't plan to do because we're just starting out. So the title reflects where we're at. We don't know the answer to this question, but this is the overarching theme of the project. And I would like to acknowledge, before I run out of time, the people that are involved in the project. My co-PI, the lead PI on the grant, is Becky Vega-Turbert at Oregon State, and the two postdocs on the project are Jesse and Joe here, and they'll be presenting a poster tomorrow. But this is a very, very large collaborative project. Working with corals is extremely challenging, both in the field but also logistically, because they're CITES protected organisms, and because of that, taxonomy is complex. So we have worked with dozens and dozens and dozens of people around the world that I don't have time to acknowledge, and some of them maybe I'll be able to mention throughout the talk. So I want to just say that this is not just this small group, but it is a global effort. Okay, so thanks all of you that have sort of set up the talk for me. You have introduced coral reefs earlier. They're the most biodiverse tropical benthic ecosystems in the planet, and people love going to them. You are familiar with the macroorganisms in these ecosystems, the fish, the invertebrates, and of course the corals. But the fundamental unit of a coral reef is a symbiosis between an animal host, cnidarian. Actually, I should use this mouse here so everybody can see. So this, what you see here is the animal. It's a colonial organism in general, and you see multiple individuals. These are called polyps, and they are basal metazoans. And in their endodermal tissue, you see here a cross-section, zooming in, where you see the endosymbiont. So these are dinoflagellate algae. The vernacular term is zooxanthellae, so people refer to them as zoox sometimes, and they belong to the genus Biodiversity. That turns out to be quite diverse. And they're photosynthetic organisms that provide photosynthate to the coral host that enables the coral to precipitate the calcium carbonate that builds the three-dimensional structure that is the city where all these organisms live in. So something that has only emerged as important in these microbial symbiosis is the other associated organisms. And this has been known as become known as the coral horobiont. So this is sort of a term that is a catch-all that refers to both the host and the Cymbidium, but also everything else associated to the system, and that includes bacteria, archaea, fungi, whole slew of protists that are unknown, phages, and viruses. And our effort is to try to characterize this microbiome using next-generation tag sequencing, targeting different markers for all these different groups. So we're in a development phase now. We've started with the bacteria and with the Cymbidium and a little bit with the fungi, but we hope to expand to all the other groups to the extent that we can, to try to understand the holobiont biology. Okay, so you've heard about mammal microbiome and plant microbiome, and we'll hear more about that, I think, later on today and tomorrow. So what is interesting about corals is that they're pretty ancient. Scarabtinian corals date back to at least the Triassic, 240 million years ago or so, but they could potentially date back to the Cambrian. So this is a very old group of organisms that have been co-evolving with Cymbidium and potentially other microbes. So we are trying to explore that. We've been fortunate enough to have access to pretty decent molecular phylogenies for the corals, and we're hoping to still contribute to improving the coral tree. This is a particular phylogeny that we've been using from the Knowlton group, where they identify many clades. Okay, so what we've done is we've based our sampling scheme on these phylogenies. So what you see is these Roman numbers associated with particular clade is what is guiding how we sample. And of course, not all these taxa are represented in a particular ocean basin. So in the first year, it's been really, really fun for the group. We've gotten to go to these really amazing reef locations throughout the world to collect the different organisms that we are sampling. And every time we go in the field, we split our sample to look at different tissue compartments. So we're not just looking at the overall composition, but we're looking at the mucus layer, the tissue, and then the endolytic layer underneath the tissue. So these are some of the pictures of some of the corals that we've collected already. Maybe, what's happening? This happened before? Anyway, so this is a map that illustrates all the different target locations, and I'm happy to report that we've gone to pretty much 80% of the sites that we collected successfully, and we're able to travel with these samples back. So we have a really large collection of tissue samples now. And this is, so I'm going to show you just a little bit of data, but because we are just vamping up to send stuff out for sequencing. But in particular, I like to highlight this group of bacteria, Endocyclomonas, that seem to be bacterial symbionts. We don't know what their role really is in the holobiome, but we see two patterns within them. So this is a microbial phylogeny of Endocyclomonas, and what you see represented here by the colors is specificity to a particular host. And then in this other clay, you see a more generalized pattern. So we're hoping to be able to reveal some of these patterns throughout the coral phylogeny. And another thing that we're hoping to do once we get this sort of deep phylogenetic framework and a handle on it is to zoom into different clades to go in deeper. And I'm going to give you one example of that. So this is the Orbicella species complex from the Caribbean from clade 17. And this is a system that we've been studying in my lab for the past 10, 15 years. It's a major reef builder in the Caribbean. It's a threatened species complex. It's of recent divergence, 1.5 million years to 0.5 million years in the case of these two species. So this is Orbicella faveolata, this is Orbicella annularis, this is Orbicella franci. And there is a very good fossil record, so we understand a lot about the Caribbean phylloclimate based on the Orbicella studies. And also, this is a study site, a transect in Panama at the Smithsonian Tropical Research Institute in the Caribbean side, where we have been able to join the efforts of Don Levitan and Nancy Knowlton and tap onto their resources. So what you see here is that the three species distribute along a depth gradient, and you have annularis in pink in the shallow areas, and then franci in the deep areas. Faveolata is not very abundant. All these colonies are individually tagged, and they have been monitored for the past 20 years or so. And we have whole genome data for the host, and we've been doing reciprocal transplants from the deep to the shallow to try to, and vice versa, to try to identify genotypes that are giving us signatures of adaptation to local habitat. And we have succeeded at that. And now what we want to do is see what is happening with the microbiome and if there is a correlation between these host genotypes and the associated microbiota. So that is work in progress. Another thing that we've been able to do with whole genome data is try to evaluate metabolic complementarity and do some modeling with that. And right now we have whole genome data for several individuals of Orbicella faveolata. The most prominent dinoflagellate symbiont has been sequenced, which used to be a major hurdle because these dinoflagellate genomes are really, really, really large. But we've been able to, we know that there is codependency and we know a little bit about the interaction, but really the genomes are giving us an overall view of what's happening. We really don't know what the associated microbiome is. We have had access so far to one microbial species associated with the Symbiodinium, but we think it's a symbiont of the dinoflagellate. And what we hope to do with this project is expand the knowledge of the genomic repertoire of this microbiome, and in particular, symbiotic microbial species. So what we've been able to do, though, just with these 3 genomes, is look at an overall, have an overall view of the metabolic complementarity. And this is just one example of one particular pathway, amino acid biosynthesis. So this is a KEGG map. And I apologize for the small font, but what you need to focus on is the colors. So green represents the enzymes that are present in the particular genome. So right now we're looking at the coral host genome. So you see a lot of gaps, and I'm going to highlight a few of these regions. So for instance, in the histidine pathway, there is an enzyme missing in the coral host. You have the end product in the tyrosine and phenylalanine pathway in the host, but there is a lot of intermediates that are missing, and that's the same case for lysine. If you look at the algal symbiont, you see something similar. In this case, the tyrosine tryptophan pathway, there is a lot more enzymes present there. And when we look at the bacterial symbiont, something really interesting emerges. And we see that this particular enzyme is absent both in the coral host and absent in the algal symbiont, but it's present in the microbial symbiont. So we're very hopeful that this approach is really going to give us a lot of insight in how this endosymbiosis, really complex endosymbiosis, is working. And even though working with eukaryotic genomes is challenging because they're draft genomes, and I'm sure that's crossing your mind. So here is the disclaimer. There is the chance that there some of these enzymes may fall in the gaps that we haven't filled. However, we see these patterns throughout the genomes overall. So we are fairly confident that we're onto something really interesting. And another thing that I don't have time to show you, but I'd like to mention, is that we have been modeling, my graduate student has been modeling metabolic flux in these interactions. And what we see is fascinating, and that Symbiodinium, in the absence of the bacterial symbiont, is actually more detrimental to the host than with it. So the algae need the microbiome to be able to actually to cooperate with the host. And in the absence of microbial symbionts, they actually be more parasitic than not. Okay, so one of the things we have the great fortune of having Jesse in the team, and Jesse is a pretty skilled computational biologist. He developed PyCrust. He was one of the early developers of Chime, too. So he developed PyCrust, which is an algorithm that uses ancestral trait reconstruction to use tag data, 16S tag data, to infer the genomic repertoire of branches that are missing or that don't have a genomic information. So this is a snapshot from the benchmarking of that algorithm, where they used all the finished genomes to test if the algorithm would be reliable, and they were able to predict sort of gene family repertoire with a pretty high confidence, 95% confidence. So what we are hoping to do is contribute to the finished genome database from the microbial isolates that we have for some of our coral hosts, so that we have coral-specific genomes. Okay, and this is what it's dashed because it's not available yet, but it will be. We then want to integrate metabolic network reconstruction in high throughput based on tag information from microbiomes. Okay, so one thing that we've been very efficient at is our reach and broader impacts, and I want to share some of that with you. So there's 3 things that I like to mention. The first one is, we'll see if the videos work. Okay, so we have already an interactive map available. And what you can do is zoom into all the reef locations that we've been to, and actually all the specimens that we've collected are cataloged in this interactive map, and you can see the reef location and a lot of the taxonomic information associated with it. That it's really crucial because coral taxonomy can be really, really challenging, and we hope to eventually link all the microbial information to the interactive map, and use the things like biospace from Illumina to share in an open access format to the scientific community our findings. So this is work in progress. The map is already available, and as soon as we start getting marker data, that will be also included. Okay, so we've done a lot of work in the Great Barrier Reef, obviously. That's where the highest diversity is, the largest reef ecosystem. So Joe has been leading this effort. It's an outreach initiative to work with indigenous children from Queensland. Another video, you're going to be only see a portion of, but this it's in its 4th year, and it targets high school students. There is a lot of indigenous knowledge that is being lost or not shared with the conservationists and the scientists that are making decisions on how to manage and protect the reef. And this is trying to bridge this gap, this educational effort to reach the gap between the Aboriginal communities and the science that is being done there. And the first group of kids have graduated from college, and the performance has increased tremendously, and a lot of them are choosing science careers or even careers in marine science. And finally, we've teamed up with an award-winning documentary film crew at Oregon State that is making a video, and they come along with us on our trips to document not only the research but also the local communities and their interactions with the reef. So you're seeing snippets. This is Saudi Arabia, the Red Sea, and this is Jesse and Ryan, the grad student at Oregon State, and they've been doing a really nice job at not just portraying the science, but engaging the local communities and local leaders to be part of this movie. And that's Becky, who couldn't make it. And this list is not complete. I'd like to acknowledge all the people, and these are some of the institutions. We couldn't fit them all in. The funding obviously comes from NSF, but recently we were able to also convince there's Microbiome project that this effort was worthwhile funding, and they're going to sequence 3,000 plus samples. With that, I'll be happy to take questions.